Yibo Zeng?
Yeah.
Our next speaker is Yibo Zeng from the Rosalind Franklin Institute, and she is going to be talking about post-translational mutagenesis, new discoveries in protein damage and repair. Yibo, the floor is yours.
Okay, thank you. I'm gonna share my screen first. Okay. Good afternoon, everyone. Today I will talk about a very interesting part of catalysis, biocatalysis. We focused on studying the two enzymes in our biological systems to see any interactions between them. First of all, let's have a look at the first protein we are looking for, human kinases protein. They form one of the largest human genome in our biological systems. They can form 3 different signal cascades to transfer the signal from extracellular membrane through the cascade reaction to the nucleus to control the expression of the proteins, cell death, migration, and cell growth. In normal circumstances, all these kinases proteins in these cascades, they have an active site containing the hydroxyl groups on the serine or threonine residues. And these conditions, they are inactive. But if they are activated by phosphorylation reactions, the phosphate group can be added to the active site. In some interesting conditions, scientists find that these hydroxyl phosphate groups can be delivered or cleaved, truncated to form the double bond dehydroamino acid short here. We give them a name, eliminons. So what's the function of the eliminons to this whole protein, whole enzyme? We have different opinions. Like shows here, Buffalo group published a paper. They claim that this eliminon can inactive the enzyme activity, but we have a completely different opinion. Why? First of all, the formation of the eliminons through 3 different pathways: protein damage, aging, the most important are by the bacterial infection. Here shows the example of the bacterial salmonella to use a type 3A secretion system to inject their virulence factor. Here shows the lias, it's a hydratase to manipulate the activities of the enzyme. So we propose that this eliminon can upregulate the kinase, the enzyme activity, which is the main cause of different diseases. For example, cancer, diabetes, inflammation, etc. In order to prove this, first of all, we have to, using chemical method to site selectively to install the DHA, dehydroamino acid, in the active site of the enzyme. Here shows through the 3 steps reaction, we first of all, to change the active site to cysteine residues. Through these reactions, using this small compound, we can change this site to the dehydroamino acid site. And in our group, former group member, Dr. Gallen, designed different derivatives of this compound. They contain different reactivity, hydrophobic, and steric and charges in order to facilitate the elimination steps to form the DHA, dehydroamino acid. Before we do the modification reaction, we noticed that there's a problem because in this native wild type and the native conditions, this enzyme has 6 cysteines in it, which will interfere our modification reaction at the active site. But if we change all these cysteines to serine residues in active one, it will completely change the structure and the function of the enzyme. So we have to find another way. So when we do the computational modification calculation to calculate the side chain accessibilities, we find that some of the cysteines, like shows here, we can change them, but there's no impact for the function of the protein. But there's one cysteine give us biggest trouble. This cysteine 207, it's very important for the function of the enzyme because it's in the ATP binding site. So we have to keep it. But how to mask it to finish our modification steps? we find that if we add the nucleotide ATP to this site, the accessibility of this site can drop from 22% to 0, but there's no changes for our target site. Furthermore, if we put magnesium ions to it, our target site, the accessibility can increase from 40% almost to 100%. That's a perfect news. But at this moment, when we do the modification reaction, we find we also get the byproduct come from the ATP, is phosphorylation DHA-ERK proteins. So therefore, we just use another nucleotide, ADP, to get our target modification site. And furthermore, we've also detect that if we add the nucleotide or magnesium ions through the thermal shift reaction, we find that all of these additions can stabilize the proteins. Therefore, by using this method, we successfully made 2 single DHA on our kinase protein and 1 double DHA. So the next step is to test the activities of this eliminome containing enzymes to see whether this eliminome can active or inactive the enzyme's activity. So we use the only substrate, the kinase protein as the protein cascade as a substrate to test the reaction. It is a 2-step reaction. We phosphorylate the tyrosine groups at the active site first, then the surrounding site. And this reaction can be easily monitored by the LCMS. So we can find the starting material, single phosphorylation and double phosphorylation. So we tried 18 different variants of our enzymes. For example, the Y-type ones, the fully phosphorylated ones. We changed the active site from serine to cysteines, and also this glutamic acid mutation, which was reported to constitutively active the enzyme activity, the eliminome form of our enzymes, the N-terminal truncated form, which is a docking region for its substrate. And we find out that for the Y-type protein, the native state, the enzyme has no activity shown here according to the reaction constant. But if we phosphorylate them, and we find that the reactivity of the enzyme increased dramatically. So that means the enzyme fully activated. But how about the DHA containing form? We find that the binding constant increased more than 20%. That means the protein, the enzyme is activated, which confirmed our suspicion. Furthermore, because of this dehydroamino acid is the electrophilic activated one, so we're thinking if we could add some nucleophilic small molecules to it to see what any changes for the activity. So this time we choose the gluphal. Gluphal is the antioxidant in our biological system. It is the highest amount of small molecules in our body, contains around 12 millimolars. We managed to add this gluphal to the DHA. We find that the activity of the enzyme dropped more than 2 times, suggesting that the addition of gluphal to this site, active site, can inactive the dysregulated enzyme activity. So at this moment, we're thinking, if there's any other enzymes, in our biological system can trap and eliminate these activated eliminomes in kinase proteins. Interestingly, at the same time, I'm a collaborator from the United States. They solve a crystal structure of human lanthionine synthesis-like proteins. These LanCL family proteins exist in various organs and tissues in our body, predominantly abundant in our brain and heart. But ever since the discovery of these family proteins 20 years ago, scientists still try to find the function of them. First of all, let's have a look at the crystal structures. When they compare with their biological homologs, we find that they share quite similar crystal structures containing 14 helix and forms 2 layers of barrier. The function of these bacterial cyclists are they can catalyze the addition of adjacent file groups to the double bond here to form the LanCL, which is the antimicrobial peptide. And we're thinking that whether our human proteins has a similar function to add a file to the double bond. When we have a close look at human LanCL proteins of the active site of them, we find that there's zinc ions in the center and 3 amino acid residues and 1 glue file can bind this to this zinc ions to form a tetrahydrogeometry. And when we compare the sequence of our human proteins with bacterial, we find that all these active site amino acids are conserved. They all showed in the bacterial and human proteins. And furthermore, all these histone residues is very important for the formation of the intermediate, also conserved in all these proteins. So we're thinking that we want to find any proteins can bind to the human LanCL proteins to finally figure out the function of them. So we stabilize these LanCL proteins on a resin and get a culture of the human mammalian cell culture to get all the proteins to pass through the resin to see any of the proteins can bind to them. Through this pull-down assay and the Proteinomics assay, we found that lots of different proteins can directly or indirectly bind to the LanCL proteins, and which include the 2 kinase proteins we just talked about. Furthermore, when we have a look at this bacterial protein cyclase, it's from the structure, we can see that it's a biofunctional cyclase, contains a dehydratase domain and a cyclase domain. This cyclase domain has a similar structure as our LanCL proteins. And this dehydratase domain shares similar patterns as our human kinase proteins. So we're thinking that there's an intramolecular interactions between these kinase proteins and our LanCL proteins. So furthermore, how about the kinases containing the DHA proteins? We also do the pull-down assay to see if any interactions between them. From Western blotting, we can confirm that this band belongs to the interaction between these 2 enzymes. Then we add the glue file to the DHA MAPK proteins. Then in the presence of the LanCL proteins, any addition could happen. Through the LC-MS, we can find that for the in the samples, we add the LanCL1 and or LanCL2 proteins. We can easily observe the shift of the peak from DHA to completely to glue file addition form. While this shifting couldn't be observed in the sample without the catalysis enzyme and also the inactive mutant enzymes. We also managed to make other DHA containing or DHB containing kinase proteins here. They share the similar patterns, similar result for the Y-type protein. For the samples we add the catalyst LanCL proteins, we can see the shift, but not in other samples. Interestingly, we also find the stereo selectivity for the LanCL proteins. We use a different DHA or DHB binding peptides and protein to test them. First of all, we choose 3 different peptides. They contain various number of DHA, DHB, or LanCL groups. We find that the LanCL cannot catalyze the addition of glue file to all these dehydroamino acid peptides. They only choose the DHB or DHA far away from the bulky stereo hindrance. So there's a stereo selectivity to the enzymes. We also find that this phenomenon, this result in the DHA containing proteins, not only in the peptide, but also in the proteins. We managed to set up a double DHA kinase protein, which contains the DHA at the active site or the random site. We find that the addition to the glue file to the DHA happens only in the Y-type proteins, and also only 1 glue file can be added to it, but not on the DHA far away from the active site. Furthermore, We also detected the regional selectivity of these enzymes to the DHA or DHB residues. For example, in the DHB containing peptides, we noticed that all these GUFA were added to the peptide in a D configuration rather than the L configuration. Recently, our group member also working on to expand the substrate pools of these enzymes. Here shows we're using the N-acetylcysteamine to add to different protein models, for example, DHA containing histone H3 proteins. Without the enzyme, the addition to the DHA should be 50-50% in D form or L form. But with the enzyme, all the addition were found in the D form. No peak shows in the L form, which will be very important for our future modification of the enzyme because it is very hard and rare to modify the protein in the D stereo center than the L stereo center. Finally, our collaborator also constructed a brilliant animal model. We knock out, delete all the LanCL genes in this triple knockout mice model, and to see any differences between these two proteins. Like shows here, for the knockout mice, we're using the mouse embryonic fibroblast cells. There's no LanCL protein can be found in the triple knockout mice. Then also, if we incubate this cell lysate with the DHA containing kinases, no GUFA can be added to the DHA in the triple knockout sample, suggesting that the LanCL protein is the only protein in our biological system which can trap and control the dysregulated DHA containing enzyme protein activity. Unfortunately, we didn't find any of the differences between these no differences or phenotyping between these two mice. And also we get the brain and the heart samples to test any differences of the protein expression. Unfortunately, we have didn't detect any of these differences. But during the pandemic, with the unknown stimulus to the mice, we observed abnormal premature death of the triple knockout mice with 6 months age, suggesting that these LanCL genes are very important for the survival of the organisms. So the summary, first of all, we find out the damage to the proteins with the dehydroamino acid residues, which will activate the kinase activities, then disrupt and alter the cell signaling in our biological systems. Through the collaboration, we solve the functions of these orphan proteins, LanCL. They can trap and eliminate the damaged kinase through addition of GUFA to fix the problem in our biological system. And also these enzymes are very important for the survival of our organisms. And the above data was published 2 months ago. The acknowledgement, I would like to thank my supervisor, Professor Davis and Professor Hutchins, for giving me a chance to working on this interesting project. And I also would like to thank my colleagues from BJD Group and UK Catalysis Hub. And a special thanks to our collaborators from United States. That's all. Thanks for your attention.
Okay, thank you very much indeed. Are there any questions for Yibo? Where do you see the research going next, Yibo?
I have two different projects, but for this project, we want to extend the substrate pool because we haven't found any of the enzyme in our biological system which can catalyze the addition of protein modification in the D form. We want to see if the D form will have a deconfiguration of the modification will have any impact for the functions of the proteins. And we can expand the protein models to different proteins which will be very important for our future work.
Okay, and what do you think the probability of you finding the right enzymes are?
We're gonna keep on using the enzyme we are using now, the LanCL, but based on this paper, we studied the interaction between kinase and our LanCL protein. But we also find that other proteins, not only the kinases, can binding with LanCLs. So I'm thinking LanCL it's a kind of protein damage mode repair enzyme in our biological system, which haven't been reported before because we are familiar with the DNA damage repair, but few reports about protein damage repair. So,
okay.
Yeah.
Okay. Very interesting. Thank you. Any other questions for Yibo? If not, then thank you very much indeed, Yibo, for your very nice presentation. The next presentation is due